Yet another video on taxonomy. In this video, we're going to look at kingdom Animalia, and then we're going to talk about the various techniques and characteristics of animals used for differentiating and classifying the 10 Animalia phyla. Okay, kingdom Animalia. So what makes an animal an animal? Well, all animals are going to be multicellular and they're heterotrophic. You have no autotrophs here. Another main characteristic of Animalia is that they're going to be mobile at least during one part of their life cycle. Animals are eukaryotic organisms, but they have no cell wall surrounding each cell. A key characteristic of Animalia is the tissue layers that form embryonically. You either have 2 or 3 or none. We're going to look at that. Then there's 10 Animalia phyla that we need to know for the DAT, and I'm going to walk you through each of them and what makes them unique. Here are the 10 Animalia phyla you need to know. So once again, we are in domain Eukarya, kingdom Animalia, and now we're dealing with phylum. As I've stated previously, many of the taxonomic questions you're going to get on the DAT will be about these 10 phyla. So it's really important that we know, okay, what makes these unique? What's their unique characteristics, and what characteristics do they share with each other? So we're going to look at that and how we characterize these phyla in the next few videos. There's many methods of classification and distinction between the 10 Animalia phyla. We're going to focus on the main and easiest ways, and those are body symmetry, looking at tissues and germ layers, a coelom, which is a type of body cavity, embryonic development, and then the digestive tract. These key characteristics are what make each phylum unique and distinguishable from each other. There's other classifications for these 10 Animalia phyla, but sometimes they are variable even within the same phylum. So what I mean by that is the modes of reproduction, for example, will differ between species in the same phylum. So it's not like, just one phylum has one answer for reproduction when you have different organisms, different groups within that phylum showing different types or modes of reproduction. But we're going to still focus on these, and I'm going to cover each of those, but there's some exceptions, and I'm going to go over that. But just know that the main and easiest ways to distinguish between the 10 Animalia phyla are going to be these characteristics up here. Let's look at the first way to characterize life in Animalia, and that's body symmetry. There's 3 types of symmetry. You have bilateral symmetry, radial symmetry, and asymmetry, which is just no symmetry. Bilateral symmetry is where you have a mid-sagittal plane running down the midline of an organism that's going to separate it into equal left and right halves. Bilateral symmetrical animals also have a head or a superior anterior region or an inferior posterior tail. So you have a head and a tail region. If you have a head, you might have this term known as cephalization. This is a process of collecting nervous tissue in that anterior region of that animal. So it's like forming a brain. So cephalization has the root cephalic in it. Cephalic meaning head. It's just a collection of nervous tissue in a head. So most but not all bilateral animals have cephalization. We're going to look at a few key examples of that as well. The next is radial symmetry. So think of like a tire where you have a circular arrangement around a central point or central axis. And with radial symmetry, you're going to have 2 sides of the organism known as a oral side and a aboral side, which means without a mouth. So you're going to have one side with the mouth, one side without the mouth. Then again, asymmetry is just no symmetry. There's no planes of section where you could divide that organism and have symmetry on both sides of that plane. And it should be noted as well that there's some animals in the various phyla that show differing body plans depending on if they're in a juvenile or larval state compared to when they're an adult. So their symmetry can change throughout their development. So it's not just locked in as one type of body symmetry. We'll look at specific animals that do this in later videos. Alright, now let's look at a few key examples of body symmetry. Remember, bilateral symmetry is going to divide the organism equally into 2 halves, a left and a right. And we are key examples of a cephalized animal where we have a collection of nervous tissue in our head, so we're cephalized. This lobster as well also shows some cephalization. A key example of radial symmetry will be a starfish. So they have a central axis that is symmetrical. You can take this in any direction. You're going to have symmetry about that central axis. On the underside of the starfish, you're going to have the oral side, so the mouth is found on the bottom. And then on the top, you're going to have the aboral side, which means without a mouth. A good example of animals that are asymmetrical, meaning they don't have any symmetry, will be sponges. So you could take a plane of section and try to divide this sponge. No matter what you do, you're not going to have symmetrical parts. So these are asymmetrical. Two words that you might run into on the DAT are going to be about tissues. So you can either be a parazoic animal, parazoa, or metazoa. Parazoa are animals that lack tissues. So if you don't have tissues, then you're not going to have a complex body plan. So that means no organs, no nothing. So the only type of animal found within the kingdom Animalia that you need to know that's parazoa is going to be sponges, which are in the phylum Porifera. Now, everything else is going to be metazoa or eumetazoa, meaning you have true tissues. So you can have complexity to your design, you can have specialized organs, and again, this is going to be everything besides sponges. So with that being said, you've already learned two unique things about Porifera, which is the phylum of sponges. Number one, they don't have any tissues, and two, they're asymmetrical. Okay, now we get to germ layers, and this is a big topic for classifying the 10 phyla in kingdom Animalia. I'm not going to go over all the embryology and development of an animal, but I'm just going to teach you what you need to know. So during embryonic development of any animal, you're going to have separation of germ layers. Now, germ layers are going to give rise to specialized cells, which go on to make specialized tissues, specialized organs, organ systems, and make the overall organism. So every animal can either be classified as a diploblast or a triploblast. Diploblastic animals have two germ layers. They have an endoderm, which is going to be your inner layer, and then they have an exterior ectoderm. So they have an outer layer, just an inner layer and an outer layer. Triploblastic animals, on the other hand, have three germ layers. They have that endoderm, they have that ectoderm, but they have a middle layer, starts with M, a mesoderm. So middle starts with M, mesoderm. So they have three. Now, way back when we were just fertilized zygotes, okay? So we didn't have to study for the DAT, didn't have a worry in the world. If you were to take a transverse plane of section and look at the cellular composition of our germ layers, we're triploblastic animals. So you would see those three layers, endo, meso, and ectoderm. You would also notice this cavity in both diploblastic animals and triploblastic animals like us. And that cavity is going to be that digestive cavity. Always surrounding the digestive cavity is going to be the endoderm. And then superficial to that endoderm, you're going to have either mesoderm if you're triploblastic, or if you're a diploblastic animal, you're going to have ectoderm. Now tying this back into body symmetry, all radial symmetrical animals, like that starfish that we were looking at, all radial symmetrical animals are diploblastic. Now all bilateral symmetrical animals are going to be triploblastic for the most part. So that's an easy way to remember it, is that triploblastic animals are going to be associated with bilateral symmetry, and diploblastic animals are going to be associated with radial symmetry. So you don't have to memorize 4 different things, just memorize those 2 key terms that go together. So a typical question might be phrased along the lines of this. An organism expresses 2 layers, namely endoderm and ectoderm. What type of body symmetry might this organism have? So you need to go, okay, 2 germ layers is diploblastic. Diploblastic animals are always associated with radial symmetry. This animal expresses radial symmetry. Now, triploblastic animals are further subdivided into different groups based on the presence of a coelom. Now, a coelom is a type of body cavity. This is not the digestive tract. So I have the coeloms drawn out in white and the digestive tract in black. A coelom refers to the space between the mesoderm and the endoderm. So this is only triploblastic animals. And there's 3 types. The first is acoelomate, which means without a coelom, there is no coelom located in the body. The second is a coelomate, and then the third is a pseudocoelomate. So a coelomate are often referred to as eucoelomate, eu meaning true, so it's a true coelomate, has a coelom, has that body cavity completely surrounded by mesoderm. So look at this coelom that I have drawn out right here, this white area. On both sides of the coelom here, it's completely touching mesoderm. A pseudocoelomate, on the other hand, is not completely surrounded by mesoderm. In fact, it's surrounded by mesoderm and endoderm. So it's almost a coelomate, hence the name pseudocoelomate, almost a true coelom. Here's just another diagram to show the difference between a coelomate and pseudocoelomate. Now remember, a coelomate does not have a coelom, so we don't have to worry about the body cavity. Now a coelomate, or eucoelomate, has both sides, both the superficial side and that deep side touching the mesoderm, whereas the pseudocoelomate is not completely touching or not completely surrounded by mesoderm. It's the deep side is touching endoderm, not mesoderm. So that's the key difference between a coelomate and a pseudocoelomate. And again, the reason we're learning this is not to just learn a fun fact, but to learn a key characteristic that distinguishes the 10 Animalia phyla. Okay, one more thing that we need to learn here about embryonic development is going to be the blastopore. So during fertilization, you're going to have a sperm and an egg, both haploid, fused together to make a 2N zygote. Now this cell, this zygote, is going to start to undergo mitotic division. It's going to start to divide. That's going to become a morula, which is a ball of cells. This morula goes on to become the blastula. The blastula is a hollow ball of cells, so you have a hollow opening or a cavity. This is really simplified, but after the blastula phase, you're going to have the gastrula phase. And the gastrula phase is characterized by the invagination to make the gut tract. So again, this is before we have to worry about endo and ecto and mesoderm. Before we even have the gut tract, we need to make it. So that's going to happen with an invagination of these cells, and that invagination, that point, is called the blastopore. So why is this important? Well, the blastopore, think about it, you have two openings to your GI tract. You have your mouth, then your anus. The blastopore can either become the mouth or the anus. Now, triploblastic coelomates, so now we're further dividing coelomates, not pseudocoelomates, this is just coelomates. Triploblastic coelomates can either be classified as a protostome or a deuterostome. If that blastopore becomes the mouth first, it's known as a protostome. If it becomes the anus first, part of that gut tract, it's going to be a deuterostome. And one more thing that you should be aware of, it's a little bit out of the scope of taxonomy, but I'm going to cover it anyways, just so you're familiar with it, is the type of cleavage that's associated with either a protostome or a deuterostome. You can think of cleavage just as cellular division, so when the cells divide, there's two different types. Protostomes undergo spiral cleavage, and deuterostomes undergo radial cleavage. Here's a dumb way to remember the difference between a protostome and a deuterostome. This is dumb, but it's going to work. If the blastopore becomes the mouth first, part of that GI tract, it's going to be a protostome. The way I remember this is I think profane words come out of my mouth when I'm studying for the DAT, right? So profane, protostome, mouth. But if that blastopore becomes the anus first, you're a deuterostome. The way I remember this is deuterostome sounds like dog doo-doo, anus, deuterostome. So I know it's dumb, but hey, now you're never going to forget it. Speaking of GI tracts, let's take a look at them. So this is not going to be associated with just triploblastic coelomates. This is going to be for all types of animals found in the kingdom Animalia. So there's two types of digestive systems in animals. Number one is called an incomplete digestive tract. This is often referred to as two-way digestion. Number two is a complete digestive tract, and this is referred to sometimes as one-way digestion. A complete digestive tract is going to have both a mouth and an anus. So food is going to travel one way down the GI tract. It goes from your mouth to your anus. This tract can also be referred to as an alimentary canal. Two-way or incomplete digestive tracts, on the other hand, do not have an anus. They only have a single opening or mouth for food. So that means they take in their food, digest it in a space called the gastrovascular cavity, and then they expel the byproducts of that through the same opening, so through the same hole. Kind of nasty. And this is used to classify, again, the 10 Animalia phyla. Okay, one last thing before we jump in and look at the 10 Animalia phyla. Let's talk about reproduction. I'm not talking about the birds and the bees here. I'm talking about the forms of reproduction. So why should you know this? If it's highly variable even within species, well, these words might show up on your DAT, so they're good to know. So for asexual reproduction, we've already looked at kingdoms that have asexual organisms, or species that reproduce sometimes asexually, like kingdom fungi. So asexual reproduction, different types of that could be budding, fragmentation, parthenogenesis. This is a big one. That's where an egg develops into a new individual, new offspring, without male fertilization. So males are not required for the species to survive. There's a few types of sexual reproduction. Remember, sexual reproduction involves two parents here. That's where you're going to have a sperm and an egg. You have two gametes, both haploid, fusing together. If you ever hear external fertilization, that's where the gametes are released into the external environment where they meet and then fuse. Internal fertilization is probably what you're thinking. That's where the gametes are deposited inside the organism, and that's where fertilization occurs. You might encounter the term monoecious or hermaphrodite or hermaphrodism. That's just referring to individuals that have both male and female reproductive organs. A species is dioecious, or sometimes referred to as sexual dimorphism, when they have either male or female reproductive organs and produce only one type of gamete. So we, as a species, are sexually dimorphic.